Hello students, today we shall learn about Anatomy of Flowering Plants, Class 11, NCERT Exercise Questions and Answers, part 1. So watch the video till the end. Welcome back once again in your biology class. This is Nowsin and you are watching your most favorite channel, Educational Support. So my lovely students, are you ready? So let's get started. Question number 1 is, state the location and function of different types of meristems. So meristems are basically 3 types on the basis of their locations in the plant body: apical meristem, intercalary meristem and the secondary meristem. So apical meristem, it is present at the apex of root and shoot and is responsible for the increase in length. Intercalary meristem basically present at the bases of leaves above the nodes or below the nodes and is responsible for elongation for the organs. And secondary meristems present on lateral side and is responsible for increase in girth or diameter. Question number 2 is, cork cambium forms tissues that form the cork. Do you agree with this statement? Explain it. So yes, I agree with this statement because cork cambium cuts off cells both on its outer side and the inner side. You know that. So the cells cut off on outer side form cork and cells cut off on inner side form secondary cortex. So the cells of cork are dead whereas those of secondary cortex are living. So I agree with this statement. Question number 3 is, explain the process of secondary growth in the stems of woody angiosperms with the help of schematic diagrams, which is its significance. So secondary growth is the formation of secondary tissues from lateral meristems, and secondary tissues are formed by 2 types of lateral meristems: vascular meristem and vascular cambium and cork cambium. So the cambium present between the xylem and phloem in vascular bundle, it is called the fascicular or interfascicular cambium. You know that. So besides this, some cells of medullary rays also become meristematic and this is called the interfascicular cambium. You know that. And both this cambium collectively constitute complete ring of vascular cambium. You can see it in this picture. So this ring of vascular cambium divides to cut off cells both on inner side and as well as for outer side. So the cells cut off on outer side are secondary phloem and inner side are secondary xylem. So the structure of secondary xylem and secondary phloem is similar to that of primary xylem and primary phloem. So with the increase in secondary tissues, the primary xylem and the primary phloem get crossed. The ray initiates of vascular cambium ring divide by tangential divisions and add new cells. So these new cells basically produced on both the sides of ray initials remain meristematic for some times and then differentiate into parenchyma cells of rays. You know that. So these parenchyma cells and rays produced by the vascular cambium in the between of secondary xylem and secondary phloem that are called secondary medullary rays. So these are usually one of few layers in thickness and 1 to several layers in height. So actually this is a figure of stages of secondary growth in a woody stem. So next is significance of secondary growth. So you know that secondary growth adds to the girth of the plant and it provides support for increasing weight of aerial parts, and secondary growth produces a corky bark around the tree trunk, and secondary growth adds new vascular tissues for replacing old non-functioning one as well as for meeting increased demand for the long distance transport of sap and organic nutrients. So these are the significance of secondary growth. Question number 4 is draw illustrations to bring out the anatomical difference between monocot and dicot root and monocot and dicot stem. So difference between monocot root and dicot root are illustrated in the following figure and the following table. So in monocot root, cortex is comparatively narrow, but in dicot root, cortex is very wide. And in monocot root, passage cells are generally absent in monocot root, but in dicot, passage cells are generally occur opposite to the protoxylem point. And in monocot, pith is either absent or very small, but in dicot, pith is well developed. You can see in this picture. Next figure is, so in monocot root and dicot root, cortex basically comparatively narrow and endodermis is less thickened and Casparian stripes are more prominent, but in dicot root, latter become highly thickened. Casparian stripes are visible only in the young root. And pericycle in monocot root produces lateral roots called cambium and part of the vascular cambium. But in dicot root, pericycle basically produces lateral roots only. Next, differences between monocot and dicot stems are illustrated in the following figure and the following table. So in monocot stem, vascular bundles basically scattered, conjoined, collateral, and bundles is usually present, phloem parenchyma absent, and xylem vessels arranged either in Y-shaped or V-shaped manner. And in monocot stem, pith is absent and ground tissues is not differentiated into the cortex and pith. You can see in this picture, and endodermis is absent and pericycle is absent. But in dicot stem, vascular bundles basically form a ring, conjoined, collateral and bilateral or open, and bundles is absent and phloem parenchyma is absent. And in dicot stem, pericycle basically made up of one or more than layers of parenchymatous or sclerenchymatous cells. And in dicot stem, pith is made up of parenchymatous cells which is situated in the center of the stem. You can see in this picture clearly. So if you dear like this video, then please subscribe my channel and please share it among the students level. Thank you for watching. Thank you for your support. Make sure you hit the subscribe button and click on the bell and stay tuned with educational support for more biology related topics. So tata, bye bye. See you in the next